Native to South America, the Humboldt penguin is an incredibly unique species of seabird. My name is Mya Daniels-Abdulahad. Come with me to explore how coastal mining is threatening these amazing birds. We begin on the western coast of South America, where Humboldt penguins occupy a region that includes the coastlines of Chile and Peru. This ecosystem is extremely valuable for biodiversity and is the focus of wide-scale conservation efforts. These efforts include the Punta San Juan Marine Reserve, which is home to the most significant Peruvian colony of Humboldt penguins. Despite extensive conservation work, the Humboldt penguin has been a vulnerable species for nearly 2 decades. Population declines are primarily driven by human activities, also known as anthropogenic effects. These include overfishing, habitat destruction, and mining. With over 200 mines nationwide, Peru is the world's second largest producer of copper, zinc, and silver, and generates a sizable amount of gold and iron. The country's largest iron mine is within 20 kilometers or 13 miles of the Punta San Juan Reserve. Contaminants from mines can travel via rivers and streams directly to the coast, where they are taken up by marine plants and animals. As contaminants work their way up the food chain, we see an effect called biomagnification. This is when metals become more concentrated in high-level predators like penguins, seals, and sharks. These organisms can also experience bioaccumulation, which is when contaminants are accumulated within the animal throughout its life. Contaminants from mine runoff include harmful compounds like heavy metals. When biomagnified and bioaccumulated, they can cause toxic effects. Heavy metals are particularly damaging to organs that clear toxins like the liver and kidneys, or tissues like fat, which can retain metals like lead and mercury long-term. This can result in liver and kidney damage, neurologic issues, and even death. Contaminants can also be transferred by mother birds into their eggs, which can cause serious harm to the developing embryo. From weakened eggshells to birth defects, contaminants cause an overall reduction in chick survival. And this makes our little chicks quite sad. Since developing chicks are some of the most at-risk individuals for exposure to environmental contaminants, it's important that we assess these threats. And since our long-term goal is to protect vulnerable penguin colonies in Peru, we didn't want sample collection for this study to reduce the number of healthy, fertile eggs contributing to wild populations. Instead, we opted to sample eggs from penguins who had been retired from the Brookfield Zoo's breeding program. Eggs were separated into shell, yolk, and albumin, also known as the white of the egg, and tested for concentrations of toxic compounds like mercury and arsenic. In shell samples, we found notable concentrations of cadmium and lead. Both of these metals replace calcium during shell formation, which weakens the shell, making it more susceptible to accidental breaks during incubation by parent birds. In the albumin, we found that metals tended to accumulate due to the presence of 2 proteins, ovotransferrin and ovalbumin. These proteins combine to useful metals like iron, forming complexes that offer protection from things like bacteria. But they can also bind to toxic metals like mercury, copper, and manganese. The key takeaway, though, is the wide range of toxins found in the yolk, which is a direct result of the way that the yolk is formed. The fats that make up yolk include 2 compounds: yolk-targeted very low-density lipoproteins, or VLDLYs, and vitellogenins. These are both generated in the mother bird's liver, and remember, the liver is a major site of toxin elimination, so it's here that toxic metals can bind to these fats, which then go on to create the yolk. Since yolk is the nutrient source for the developing embryo, metal contamination in this part of the egg can have critical effects on the chick's development and health. So why is this research important? These data confirm that toxic metal compounds are deposited into Humboldt penguin eggs. While the concentrations found in this study weren't high enough to cause direct harm to a developing embryo, it's important to keep in mind that we collected eggs from birds in the zoo who experience lower exposure to environmental contamination than wild birds would. We can therefore expect that metal concentrations in wild birds would be significantly higher. Additionally, birds act as sentinels of environmental health. Since Humboldt penguins hunt in the same coastal areas where fish are harvested for human consumption, the detrimental effects we see from metal exposure in penguins may also be seen in humans. This applies to environmental contamination as a whole, as heavy metals from mines can easily contaminate local waterways and agricultural areas, which would then contaminate crops and livestock. As surprising as it sounds, by establishing the effects of contaminants on Humboldt penguins, we're able to better understand how human health and environmental conservation are intimately linked. The findings of this project support the need for ongoing conservation work in Peru to address the negative impacts of mining. To protect the Humboldt penguin, as well as all of the other wildlife that call this region home, we must continue to preserve coastal habitat, advocate for sustainable mining practices, and educate the public on how to best support the recovery of affected marine ecosystems. With our continued efforts, we can start the wave that creates a sea of change in the conservation and protection of Humboldt penguins living in Peru. These findings were published in the Journal of Wildlife Diseases. I extend my gratitude to our collaborators, supporters, and my co-authors for their work on this project. Thanks for watching!